Hello everyone, let's get started. Today we're going to be talking about the cell cycle and cell division. On this slide, I've got an overview of how we're going to approach this topic. First, we're going to define exactly what the cell cycle is. We're going to look at the different stages, including a comparison of the M phase that occurs in both somatic cells and reproductive cells. In other words, a comparison of mitosis versus meiosis. We'll talk about what a generation time is and how generation times vary between cells that are living within an organism and cells that are being grown in the laboratory. We'll look at some of the differences that we see in embryos. We'll talk about the cell cycle checkpoints, why they exist, and what they do in order to regulate the cell cycle. And we'll finish up by thinking about some of the problems that can result when cell cycle checkpoints go awry. But we'll begin today just by defining exactly what the cell cycle is and what stages are within it. Remember, cell division begins with what we call a parent cell. It ends when the parent cell divides to produce what we call daughter cells. The cell cycle can be thought of as containing several different individual stages. We give each one of these a name. It begins with what we call G1, then moves into S, then into G2, then into M, and it finishes with a process called cytokinesis, which is the physical division of the parent cell into two or more daughter cells. There is one other stage that we talk about with regards to the cell cycle called G0 or G0. Let's begin with G1. G1 stands for gap 1. This stage got its name because when the cell cycle was first being investigated in the laboratory, scientists noted that during certain stages, no activity could be seen in the cell underneath the microscope. The stages where nothing seemed to happen were called gap phases. So during the first gap, while it looks like nothing's going on, a lot is actually going on. This is the time when the parent cell begins to replicate everything that is inside. The cytoplasm, all of the dissolved proteins, the organelles, everything that is within a parent cell has to be doubled so that when the parent cell is physically divided, each daughter cell gets a full complement of all the materials that are necessary for life. During the first gap, all sorts of materials will begin to be replicated, with one exception, and that exception is the DNA. The DNA molecule is not replicated until we move into the next stage, which is S. S stands for synthesis, and it's during the S phase that the DNA molecule is replicated. Remember, during DNA replication, the entire DNA molecule is copied so that each daughter cell gets one full copy of the genetic material. After the S phase, we move into G2 or gap 2. This is very similar to gap 1 in that it is a continuation of the processes that replicate all of the materials in the cell, with the exception of the DNA. After G2, we move into the M phase. Now, students often think that the M phase stands for mitosis. In reality, M means nuclear division. This is the stage where the nucleus, which now contains 2 full copies of DNA within it, this is the stage where the nucleus will be divided into 2. It's important to remember that M does not stand for mitosis. It stands for nuclear division. And that's because it can take 2 different forms. Mitosis is what M phase looks like in some types of cells, but in other cells, M phase takes the form of meiosis. Lastly, that additional stage of the cell cycle that we called G0 is the stage that a cell enters into when it leaves the cell cycle for some reason. So whenever a cell enters the cell cycle, there will be times when, for different reasons, it has to exit the process of cell division. When it does, we say that it enters into G0. Now, the other terminology that we need to know regarding the stages of the cell cycle is that we can combine G1, S, and G2 together into what we call interphase. So when scientists were first examining dividing cells under the microscope, they noticed that very little was happening, very little was visibly happening during those first 3 stages, G1, S, and G2. It wasn't until the cell moved into the M phase that these early scientists were able to actually see things happening. So they grouped G1, S, and G2 into the term interphase. On this slide, we're looking at a diagram of the cell cycle. The cell cycle is often drawn this way, looking like the face of a clock, because it does depict a cycle. Dividing cells don't just divide once, they divide over and over again during the time of their life cycle when division occurs. So we draw this process as a cycle, repeating around and around again. It starts with G1, moves into S, then into G2, and then into M. Remember, in some cells, M, or nuclear division, will take the form of mitosis, and in other cells, it will take the form of meiosis. You'll notice that G0 is drawn off to the side. G0 is the stage that cells enter into when they leave the cell cycle. G1, S, and G2 together are referred to as interphase. So let's take a minute and compare the M phase between cells that use mitosis and cells that use meiosis. What mitosis is a form of nuclear division that produces 2 genetically identical daughter nuclei from one parent nucleus. So again, it's important that you remember that M is about the nucleus, not the entire cell. During M phase, the nucleus is divided. The entire cell is not divided until the cell cycle ends, until M phase is complete and the process of cytokinesis occurs. During M phase, if the cell is using mitosis, the parent nucleus will be divided into 2 genetically identical daughter nuclei. In other words, the DNA inside each of the new daughter nuclei will be exactly the same as the DNA that was in the parent nucleus. It's interesting to note that Mitosis occurs in all cells at some time during their life. So, of course, we're only talking about eukaryotic cells here, because only eukaryotic cells have a nucleus. But even in a human being, all of the cells in the human body go through mitosis at some point during their lifetime. So even cells that will move on and start to use meiosis perform mitosis at least at one point in their lifetime. The reason that a cell uses mitosis is to assist in the growth process, and by growth we just mean increasing cell number, to assist in tissue renewal, and by renewal I mean simply replacing old worn out tissue with new cells, and finally tissue repair. There are times when tissues get damaged and we need the cell cycle and mitosis to occur in order to repair that damaged tissue. Meiosis is a very different process, and meiosis occurs in a much smaller subset of eukaryotic cells. Meiosis is a form of nuclear division that results in the production of haploid gametes. Now, I've written the number 4 here, and I've got a little asterisk next to it. I will explain that in just a minute. Meiosis produces 4 haploid gametes from a single diploid parent cell. So we need some definitions to help us understand this explanation. Remember, we're talking about eukaryotic cells here, and eukaryotic cells contain linear chromosomes. When we say that a cell is haploid, what we're saying is that it contains one of each type of linear chromosome. When we say that a cell is diploid, the DI translates as 2. So we're saying that this cell contains 2 of each type of chromosome. If you use human beings as an example, human cells contain 23 types of chromosomes. And since humans are diploid, we get 2 copies of each one of those 23 types. You inherit 1 copy from your mother and 1 copy from your father. So for all 23 types of chromosomes, you get 2 of each, for a total of 46 chromosomes. Now, meiosis is a process that occurs in only a small subset of eukaryotic cells, and it occurs in the stem cells that make the gametes. So these are the cells within the male gonad or testicle and the female gonad or ovary that function to produce the gametes. Meiosis is occurring for one reason only. It's occurring to produce the gametes, the sperm cell, the egg cell, that are required for sexual reproduction. So meiosis is only occurring in eukaryotic organisms that use sexual reproduction. Now, let's take a look at the number 4 up here. In reality, in a male organism, 4 haploid gametes will be produced through meiosis from a single diploid stem cell in the testicle. It's different in the female. In the female, even though the process is producing 4 haploid gametes, there's some strange goings-on that occur. that eliminate all but one of those haploid gametes. So, in a male, in the testicle, for each stem cell that goes through the process of meiosis, 4 haploid sperm cells will be produced. In the female, in the ovary, for each stem cell that goes through the process of meiosis, only 1 egg cell will be produced, along with 2 other structures called polar bodies. We'll talk a little bit more about this strange process in the female in just a few minutes. Now let's go back into the cell cycle and look at the M phase of mitosis. Let's talk about the stages that are present within mitosis and what is happening inside the nucleus in each one of those stages. Mitosis contains 5 individual stages: prophase, prometaphase, metaphase, anaphase, and telophase. During prophase, the chromosomes are going to appear underneath the microscope. Remember, during most of the lifetime of a cell, the DNA exists in the nucleus in the form of chromatin. It is wrapped around histone proteins and carefully stacked into what we call compacted chromatin. When the cell enters into prophase, that chromatin is going to get even more tightly packed. That process is known as condensation, and it produces structures that can be seen under the microscope that we call chromosomes. So, chromatin is compacted, chromosomes are condensed. Now, the other event that happens during prophase is that the spindle apparatus appears. Remember, the mitotic spindle is going to emerge out of the centrosomes inside the cell. The mitotic spindle is made out of microtubules. So during prophase, microtubules will begin to grow out of that pair of centrioles inside each of 2 centrosomes. There'll be 1 centrosome that will eventually move to one end of the cell and 1 centrosome that will eventually move to the other end of the cell. Each centrosome contains 2 centrioles, and microtubules grow out of each of those centrioles. This is an elaborate apparatus that will be used to physically move chromosomes around in the parent cell nucleus. It first appears during prophase. The next stage of mitosis is called prometaphase. During prometaphase, the nuclear envelope is going to break down, and all of those newly condensed chromosomes are going to be released into the cytoplasm of the cell. The next stage is called metaphase. During metaphase, the newly released chromosomes are going to move into the center of the cell and line up along a structure called the metaphase plate. Remember, each of these chromosomes was replicated during the S phase of the cell cycle. So we're going to see each chromosome with its pair. And all of the different types of chromosomes with their pairs are going to line up head to tail along this metaphase plate, right down the center of the cell. I'll show you a picture of this in just a minute. The next stage is anaphase. During anaphase, these replicated chromosomes are going to get physically pulled apart. One set of chromosomes will be drawn toward one end of the cell or one pole of the cell, and the other set of chromosomes will be drawn towards the other pole of the cell. The last stage is called telophase. During telophase, a new nuclear envelope will be produced around each set of chromosomes at each end of the parent cell. On this slide, we're looking at drawings that represent each of the stages of mitosis. So we're starting off with a cell that is in interphase. Remember, under the microscope, during the cell cycle, most of the cells that are in the cell cycle are going to look like this. If we looked at a group of cells under the microscope that we knew were entered into the time of cell division, some of them would be in G1 at any one moment, some of them would be in G2, some of them would be in S, and some of them would be in M. So most of the cells would look like this. Most of the cells would be in either G1, S, or G2, and nothing interesting would be happening, at least visually, inside the nucleus. Now, they've drawn in for us a centrosome over here. Actually, they've drawn in two centrosomes. Remember, inside each centrosome, there's a pair of centrioles. Now the cell is going into M phase. This is a cell that is going into mitosis as its M phase. So during prophase, remember, two things are happening. The chromosomes are going to condense, and the spindle apparatus is going to appear. Inside of the nucleus, we can see these linear structures appearing as the chromatin condenses and produces these chromosomes. Outside of the nucleus, we can see that the centrosomes are moving towards opposite ends of the cell, and out of the pair of centrioles inside each centrosome, microtubules are being constructed. The spindle apparatus is being assembled, it's being readied. The next stage is prometaphase. Remember, this is one that's very hard to catch under a microscope because it happens quickly. During prometaphase, the nucleus essentially breaks apart. The nuclear envelope is taken down, and the newly formed chromosomes are released into the cytoplasm. During metaphase, those chromosomes are going to line up across the center of the cell. They line up head to tail. Next comes anaphase. During anaphase, each set of chromosomes gets pulled to one end of the cell. So one set will head to this side of the cell, the other set will head to this side of the cell. It's the microtubules that are pulling apart the chromosomes that lined up along the metaphase plate. The last stage before cytokinesis is telophase. During telophase, new nuclear envelopes will be formed around each set of chromosomes at each end of the cell. At the end of telophase, cytokinesis occurs. Cytokinesis, if you translate it, means division of the cytoplasm. So during cytokinesis, we're going to see the membrane of the parent cell divide down the center and eventually pinch off, producing two daughter cells. Now let's take a look at the process of meiosis. Unlike mitosis, meiosis actually involves two divisions of the nucleus. During the first division, we have early prophase 1 and late prophase 1. we have Metaphase 1, Anaphase 1, and Telophase 1. During the second division, we have Prophase 2, Metaphase 2, Anaphase 2, and Telophase 2. So this is a process, meiosis, where we're going to have not one division of the nucleus, but two. And you can see that a lot of the individual names of the stages within meiosis are the same as what we saw in mitosis, but there are some important differences. First of all, you'll notice that there is no prometaphase going on in meiosis. It's important to know that the process where the nuclear envelopes are breaking down is happening during meiosis. We just don't give it a named stage. So while we don't list prometaphase in here, it's essentially still happening. The nuclear envelopes have to break down and release the chromosomes in order for the rest of the stages to occur. Another noticeable difference is that prophase, at least in the first meiotic division, has been broken into an early and a late half. In mitosis, remember, there's only one prophase. It's really important to remember this early and late division of prophase because something very important is happening during late prophase 1. When we compare meiosis to mitosis side by side, the second division of meiosis looks very similar to mitosis. It's the first division that looks quite different. So, early prophase 1 is going to look very similar to prophase 1 of mitosis. During early prophase 1, we're going to see the chromosomes condense and the spindle apparatus appear. During late prophase 1, something very different is going to happen, and that's called crossing over. Meiosis, remember, is the form of nuclear division that helps produce the gametes in sexually reproducing organisms. And as you probably already know, there is a process that goes on in the production of gametes that mixes and matches source DNA that comes from the mother and the father, so that each individual gamete, each sperm cell, each egg cell, has some DNA from the mother and some DNA from the father. Crossing over is what produces this. Similar types of chromosomes that are inherited from the mother will cross over with similar types of chromosomes that are inherited from the father. They will switch and swap bits and pieces of DNA between them. That's crossing over, and it occurs in late prophase 1. Next comes metaphase 1. And just like the metaphase we see during mitosis, during metaphase 1, chromosomes are going to move to the center of the cell, and they are going to line up. What makes metaphase 1 different from what we see in mitosis is that we're going to see homologous pairs of chromosomes line up. So we won't see a head-to-toe arrangement, we'll see a side-by-side arrangement. And I'll show you a picture of this in just a minute. Anaphase 1 looks very similar to the anaphase that we see in mitosis, in that the spindle apparatus is going to pull chromosomes apart and move them to each end of the cell. Telophase 1 is going to look very similar in that new nuclear envelopes are going to be formed around each set of chromosomes. Remember, at the end of telophase 1, we're not finished with meiosis. We immediately enter into the second meiotic division. There will be a second prophase where chromosomes condense and spindle apparatus appears. There will be a breakdown of the nuclear envelopes, even though we don't recognize it as a stage. There will be a second metaphase where chromosomes line up in the center of the cell. During metaphase 2, though, it looks just like what we saw in mitosis. They're going to line up head to tail. Then we move into Anaphase 2, where the spindle apparatus pulls the chromosomes apart and sends them to each end of the cell. Then there will be a Telophase 2, where new nuclear envelopes will be formed around each set of chromosomes. And finally, at the end of Telophase 2, we will see 4 individual gametes produced. Now, it's important to notice that the first division of meiosis does have a cytokinesis at its end. And the second division of meiosis also has a cytokinesis at its end. This is why you end up with 4 gametes, at least in the male. One parent cell produces 2 daughter cells at the end of the first meiosis, and then 2 daughter cells each go through the second division, go through a cytokinesis, and each produce 2 daughter cells. So from one parent cell that enters meiosis 1, we get 4 daughter cells coming out of meiosis 2.